So now that you know all about the biochemical properties of the different classes of macromolecules, we're going to start talking about how these function in the cells. And in particular, we're going to start with cell membranes, and proteins and fatty acids and carbohydrate classes of macromolecules will be particularly important for this, this part of the course. So with regards to the cell membrane, we'll talk about different structures of the cell membrane, and we'll also talk about the function of different components of the membrane. So in particular, you should be able to answer a few questions when we're done with this section. The first one is, what are the parts of a membrane? So this includes the phospholipid components as well as other protein components and carbohydrate components. You should be able to talk about what experiments scientists did perform to determine the structure of the membrane. We'll talk about how components of the membrane interact to give them a particular orientation or structure, and importantly, what functions these components perform based on their structure. So I'm going to give you a few examples of how dynamic the cell membrane is. The first one is this macrophage, which has detected a foreign body, and so it sends out these processes. These are bacterial cells in green. And the outcome of this is that the macrophage can then pull the bacterial cells back in, endocytose them, and then digest them as part of the immune response. So this is a great example of how flexible the cell membrane is, and how important cell signaling can be as the macrophage has detected something that indicates that this bacterial cell is a foreign object. This is another example of a macrophage in pink, and these blue spots are false colored to show you virus particles that are escaping from the macrophage or being shed. And so as these particles are shed, they'll take small pieces of the cell membrane with them. You can see a small one that looks like it's coming out right here. So this will create a small hole in the plasma membrane, and yet these holes seal up very quickly because of how fluid the cell membrane is. And this is a video from your textbook, which is showing a neuron with a little extension of this neuron. This white spot here is a laser tweezer, and you'll see it pull out a piece of the membrane, and then it can move this piece of membrane fairly quickly back and forth. So it's a really impressive example of how fluid the membranes are, and we'll talk about the components that contribute to that fluidity as well. Some of the earliest studies examining the components of a cell membrane were done by Overton in the late 1800s. So Overton showed that substances could cross their membranes at rates that were similar to their oil-water partition coefficient, and I'll explain that in a second, but that some crossed membranes faster or slightly slower than expected. So this graph is showing rate of diffusion in the membrane on the y-axis, and the x-axis is each component's solubility, each molecule's solubility in a lipid. So if you add a molecule to an oil and water mixture, then you can measure which partition that molecule tends to be found at different concentrations in. So you can see that overall there seems to be a fairly linear relationship between the rate of diffusion in a membrane as compared to the solubility of the molecules in lipid. However, you'll notice that a couple of these seem as though they are diffusing slightly slower. For example, a trimethyl citrate here appears to be slightly slower than you would have expected it to diffuse in a membrane. So Overton observed that the lipid layer was a barrier to polar molecules, but was not a barrier to nonpolar molecules. So he concluded that the cell membrane had a lipid nature, and that there were lipids on the surface of the cell. He also found cholesterol as a component of cell membranes, and that could also help account for this barrier to some of the components. So in fact, if you make an artificial lipid bilayer, so you can just mix lipids in an aqueous environment, you can observe how different classes of molecules can diffuse across that lipid bilayer. And as I mentioned, you can predict this based on their oil to water partition coefficient. So small non-polar molecules, such as oxygen or steroid hormones, can diffuse across the lipid membrane very easily. Small uncharged polar molecules generally will not cross the lipid bilayer, but if you manipulate the concentrations, you can force some diffusion across the layer. The same is true, but to a lesser degree, for larger uncharged polar molecules, so this includes amino acids, glucose and nucleosides, and ions such as sodium, potassium, chloride, etc. will not pass this artificial lipid bilayer. And so this corresponds to the observations that Overton had seen that we just looked at previously. Okay. Some polar molecules, if you have natural vesicles made from a cell membrane, will diffuse across the membrane faster than expected. So, for example, artificial vesicles, as we saw in the previous slide, could just be lipids mixed in an aqueous solution, and the rate of movement across the membrane corresponds to the concentration of the substance. However, if you make vesicles with membranes from cells, so we know now that these membranes contain various proteins, these real vesicles that are from a cell that has some membrane proteins, you'll see the rate of movement across this membrane is quicker than if you just have artificial vesicles with no proteins. So this linear line is showing us the rate of simple diffusion. So this is the characteristic of the substance that depends on its water-oil partition coefficient. However, you may have heard before of facilitated diffusion, and that's what we're seeing in this blue line, where real vesicles have a faster rate of movement across the membrane. So we'll talk about the different components of the cell membrane that contribute to each of these observations. And we'll slowly build how the Singer-Nicholson fluid mosaic model of membrane structure was established. So if you have taken a course that talked about cell biology before, you probably know a little bit about how this mosaic model illustrates the cell membrane. So we have the phospholipid bilayer, we have glycolipids and proteins that are in the layer of phospholipids as well as associated with the phospholipids. And this is the current accepted view of membrane structure that we will talk about how it was developed and what experiments along the way proved ultimately that this is the most likely explanation for the structure and function of cell membranes.